Hi, everyone. My name is Luis Rodrigo Arce, and I'm going to talk to you about our research testing the predictions of reinforcement, long-term empirical data from a disulfide hybrid zone. So, as you may know, reinforcement theory explains how speciation can occur in sympatry due to the negative selection of maladaptive hybrids. So, while hybrids are negatively selected, selection will act to increase the initial differences between the two species in sympatry. Over time, this may cause that the variance between the two species in sympatry becomes higher than the two species in allopatry, a pattern called reproductive character displacement. Over time, this may lead to the completion of reproductive isolation and thus to speciation. This reduction in hybrid fitness is usually explained due to BDM incompatibility model. These are alleles that originated in allopatry and that causes a reduction in hybrid fitness when they are found within a hybrid genome. Usually, these BDM incompatibilities are found within autosomal chromosomes. However, they can also be found in sex or in mitochondrial sex chromosomes or mitochondrial DNA. And when this happens, the unidirectionally inherited processes of this DNA causes differences in the hybrid fitness or polyzygotic isolation between hybrids formed in the two crosses direction. Since there are differences in the polyzygotic isolation of these hybrids, reinforcement processes may act also differently, causing also differences in the prezygotic isolation between these two crosses. And although several reports have been made of these asymmetric reinforcement processes, it is still unknown how frequent do these processes occur in several localities of the same pair of species. Mosaic hybrid zones give us an opportunity to test this by sampling several contact regions between the two parental species, and we can measure reproductive barriers and then test the consistency of these asymmetrical reinforcement processes. One model that has become a very interesting model to study hybridization and speciation are the Ischnura elegans and Ischnura graelsi damselflies. These damselflies used to have an allopathic distribution in which Ischnura graelsi was found in Spain and Portugal, while Ischnura elegans was found from France to Eastern Europe before the 1900s. However, due to climate change, Ischnura elegans has had a range expansion and entered in contact with Ischnura graelsi, forming several sympatric regions within Spain, one of which is the western hybrid zone, found mainly across the Galician coastline, and in which there are allopatric populations of both species, as well as sympatric populations where the two species hybridize and reproduce and hybridize. So we visit the allopatric and sympatric localities of the two species across Europe, gathered individuals in the laboratory, and made experimental crosses, interspecific experimental crosses between them, and measured reproductive barriers. These reproductive barriers represent key components during reproduction of these damselflies. The first one is the mechanical isolation. This is the mechanical compatibility between the caudal appendages of the males and the pronotum of the female. If males grab correctly the female during the first step of reproduction, then the tandem position is made. So we measure it using a binomial distribution, the frequency of successful to unsuccessful tandems. Then, if the female is grabbed and stimulated correctly, she will bend her abdomen to place in contact the genitalia of the two organisms. This causes this copulation or mating position, and also we measured the frequency of this position as a binomial parameter. During copulation, males will transfer the sperm from their body to the females, in which the females will storage the sperm both in the spermatheca and in the bursa. If this transfer of sperm is made correctly, then females will have the chance to have their eggs fertilized and to oviposit. So the ratio of females that oviposit and that not oviposit was also measured as a binomial barrier. And finally, in the females that had the chance to oviposit, we measured both the average number of eggs that they laid per clutch, as well as the ratio of fertile to unfertile eggs that they laid. And we measured all these barriers both in during heterospecific crosses and in second generation crosses using hybrid samples. And finally, for each of these barriers, we measured as a reproductive isolation value that goes to 0 from 1. But also, we measured the relative cumulative isolation. This is the contribution of these reproductive barriers across sequential steps of reproduction to the total cumulative isolation between these two species. So in this plot, I'm showing you the cumulative reproductive isolation of several crosses, and the colors represent different populations. The first pattern that you may see is that hybridization occurred asymmetrically and in opposite directions between allopatric and sympatric crosses. In allopatry, crosses between Ischnura elegans males and Ischnura grailsi females were completely isolated. However, in the opposite direction, we had the production of hybridization. In sympatry, in 2 out of 3 geographical repetitions, we found that the Ischnura grailsi males with Ischnura elegans females cross direction was completely isolated. And we explained the difference seen in this 3rd geographical repetition as a product of low sample sizes and the high frequency of hybrids that were in this population, and that may cause a mal-identification of hybrids and the involvement of these hybrids during all reproductive barriers measurements. However, in 3 geographical repetitions of the other cross direction, we found that hybridization occurred between Ischnura elegans females and Ischnura grailsi females in sympatry. So to test the presence of reinforcement in this system, we measured and tested several theoretical predictions that this theory has. The first one is the classical pattern expected or predicted by Dobzhansky, in which prezygotic isolation should be higher in sympatry than in allopatry. So here I'm showing you the outcomes of the mechanical barriers, in which green represents the relative positive outcome, successful tandem formation, and red, the negative outcome, the unsuccessful tandem formation. And you can see that we found statistical differences via GLM modeling in crosses between Ischnura grailsi males and Ischnura elegans females in sympatry that had a higher mechanical isolation than the same direction in allopatry. However, this pattern is not enough to conclusively conclude the presence of reinforcement, since other processes can also make these patterns, such as the Templeton effect. The Templeton effect or differential fusion stipulates that sympatric populations can only occur in nature when, since allopatry, these species had already strong differences or reproductive isolation. If differences between allopatric populations is low, then when they merge together in sympatry, they will fuse and we will not be able to see these sympatric species. However, if the two species have already a strong differentiation in allopatry, then we can see these sympatric localities. As you may see, it's the same pattern expected as reinforcement, but it's a different processes. So the other defendants of the reinforcement theory made other predictions that should be fulfilled when reinforcement was occurring. The first one is the rarer female effect that explains how females from the rarer species have a higher chance of entering in an hybridization event, and thus will be more prone to be reinforced than females from the other species. In our case, females from Ischnura elegans, Ischnura elegans is the rarer species since it is the invader species in Spain, and thus we expected the direction with Ischnura elegans females to have a higher isolation than Ischnura grailsi females. And this is exactly what we saw, and we found these differences in the mechanical barrier. And the third prediction was of a concordance between prezygotic and postzygotic isolation due to the inheritance of unidirectionally inherited BDM incompatibilities. However, we couldn't test these predictions because both in allopatry and in sympatry, one of the two crosses directions were completely isolated. Although this third geographical repetition didn't have a complete isolation in sympatry, we could not re-rob hybrids from this direction because of the low sample sizes that we made in this cross. However, and thus, since we didn't have hybrids from both directions, we could not test this prediction. The fourth prediction is that reinforcement expects greater premating asymmetries in sympatry than in allopatry. And this is exactly what we found. So here I'm showing you the differences between absolute isolation of crosses from one direction minus crosses from the other direction. And you can see how premating barriers, specifically the mechanical barrier, are significantly different between the two crosses direction in sympatry, but not in allopatry. And you may see even how the stronger mechanical isolation has shifted from the direction of Ischnura elegans males with Ischnura graelsi females to the direction of Ischnura graelsi males and Ischnura elegans females in sympatry. And finally, since while reinforcement occurs in sympatry, some introgression may occur via gene flow in sympatry but not in allopatry. Thus, this introgression may place BDM incompatibilities in light of the selection, of natural selection. Thus, we expect a purging of Bayesian-Dobzhansky-Miller incompatibilities and thus a reduction in post-zygotic isolation in sympatry but not in allopatry. And that is also a prediction that we found, since here you can see the fecundity values in these violin plots on each of the hybrid crosses, post-zygotic hybrid crosses, and you may see how sympatric crosses, the ones in pink, have higher fecundity values that are allopatric controls. And this pattern was also seen in the fertility barrier, the ratio of fertile eggs. However, this prediction may be taken cautiously because remember that hybrids from allopatry and from sympatry came from opposite cross directions. And finally, our results were consistent with recent morphological evidence made by my partner Andrea in the lab that has found a pattern of reproductive character displacement of higher variance in differences between the two species in sympatry than in allopatry, specifically in the pronotum of Ischnura elegans females. So both our reproductive barrier measurements and our morphological analysis are consistent with the key and log model of insect evolution that explains how male and female structures co-evolve to avoid hybridization. Finally, reinforcement, we hypothesize how reinforcement and gene flow may have changed hybridization directions between allopatric and sympatric crosses. We think that after secondary contact, hybridization occurs between Ischnura graelsi males and Ischnura elegans females, such as in allopatry. However, the high cost of hybridization and lower frequency of Ischnura elegans made this direction to be reinforced. However, during this process, some introgression and gene flow could have occurred, causing the purging of BDM incompatibilities. With this reduction of BDM incompatibilities, hybridization may have begun to occur in the opposite direction in sympatry between Ischnura elegans females and Ischnura graelsi females. So an interesting question for the future is, will reinforcement also occur in this direction, or will gene flow and introgression will prevail? And finally, we found evidence of the role of the X chromosome in the isolation of this species, since in allopatry or post-zygotic isolation show that every cross involving hybrid males was completely isolated because hybrids were unfertile. And this is expected because males are the hemizygous sex in this species, consistent with the Haldane rules. So, as conclusions, we have new empirical evidence of reinforcement in Odonata, consistent with the rare female effect. We show how reproductive isolation was driven by several mechanisms, such as reinforcement, BDM incompatibilities, the key and log model, and the Haldane rules, and how reinforcement may have increased isolation in one direction, while gene flow weakened it in the opposite direction. And I finally want to say thank you for all the people involved in my PhD thesis, as well as the INECOL, the CONACYT, the Evolution meeting, and all the people hearing this talk. Thank you.